It's a great pleasure. Of course, I've been to Vienna quite a few times, and the new lab that we have now, we've had it for about 8 years. When we were designing it, I did come and visit, I think 2006, when Barry Dixon had just become the head of the IMP at that point, and we were shown round and shown all the ideas, and we took all the ideas from all the different labs we visited, and then we have a new lab, relatively new, similar to the great new lab that you now have as well. Yeah, so, and then this talk was planned about 3 years ago, and so I haven't changed the title actually from 3 years ago. But nevertheless, let me start and tell you a bit about how we got to where we are in terms of a little bit about the history, and then how things are going, and then where things were going in the future. So when I started out as a PhD student from physics, I didn't really plan to become a structural biologist. I was just looking for biophysics, but I ended up doing X-ray crystallography. And that's what in 1973 or so, after being a postdoc at Yale, trying to work on membrane proteins and having identified this bacteriorhodopsin specialized part of the membranes. So this is a freeze fracture picture of Halobacterium holobium taken by Walter Stichinius, who discovered the identity of these specialized patches with one protein, bacteriorhodopsin, in it. And I'd started working on that at Yale and was trying to make purified protein and make 3D crystals for X-ray crystallography, when I heard a talk by Nigel Unwin, who had also done his PhD, but in metallurgy in Cambridge. And he had been taken on by Hugh Huxley, recruited as a young group leader to try to develop electron microscopy technology for biology using his background in material science. And I said to him that these membranes might look very good samples for looking at it in electron microscopy. So we recorded electron diffraction patterns like the ones on the top left, and images that I'm not showing you that were low-dose images, processed them and ended up with this model that Clemens mentioned before in 1975, which was a surprise to us actually, how nice and clear it was. But without working with Nigel for a couple of years, I would still be doing X-ray, trying to do X-ray crystallography. And then after about another 15 years, so 1975 to 1990, we finally got a map at about 3.5 angstrom resolution where we could identify features of the amino acid sequence, which by that point had been determined. And then we were able to build an atomic model. You can see the phenylalanine, tyrosine, tryptophan, bigger side chains, and then infer the medium and smaller side chains along alpha helices from that. And so that was going from X-ray crystallography to electron crystallography, two-dimensional crystals. And then later on, about mid-1990s, I was asked to give a talk about radiation damage for people who wanted to build an X-ray microscope in Grenoble. Did some calculations, presented a talk showing that the amount of damage by X-ray diffraction compared to electron diffraction and imaging, for each useful elastic event, that means an X-ray or an electron contributing to a nice image or a diffraction pattern, the damage by X-ray was 1,200 times bigger than electrons. And so if you wanted to look forward and try and do atomic resolution microscopy, clearly electrons were better. And then having shown that, we ended up making this attempt to do a first principles calculation to ask, what could you do if you didn't have a crystal? And you can see on the left-hand side at the bottom, there's a sort of model which is meant to be a single particle, but in order to do the calculation, we've pretended it's in the form of a crystal. And then the question was, how much information do you need from the image, if you record a perfect image, in order to find whether a molecule is there and find its position and orientation? And that's what these columns are. These two columns, this is can you find the molecule if it's a very big virus, or a ribosome, and then down to smaller proteins, eventually very small proteins. And it turns out, if you say signal to noise has to be above 3, anything above 10 or so kilodaltons, you can see it in the image above the noise level, and anything above about 30 or 40 kilodaltons, you should be able to find its orientation as well. And it was this calculation, and then the right-hand column was what we believed the number of molecules you'd need to get atomic resolution structures, which was 10,000. And it turned out we were very conservative in that calculation. It turns out you should only need about 500 or 1,000. If you use the same criterion that's used to determine whether diffraction spots in X-ray crystallography are significant or not. So it was this calculation that convinced me we should stop doing electron crystallography and start doing, in our case, other people had been, Joachim Frank had been doing single particle, but without cryo before that. So we switched our efforts from doing electron crystallography and attempting at the same time to make 3D crystals for X-ray crystallography to doing single particle cryo-EM. It was about 25 years ago now, and it turned out there were lots of problems. The microscopes didn't have field emission guns, the vacuums were bad, the detectors were bad. So we started work on it, and in the year 2000, Tim Baker and I, this is from a review in the International Tables for Crystallography, volume F, which had about 90 chapters. Michael Rossman and Eddie Arnold were the editors. They recruited 88 people to talk about X-ray and a little bit of neutron diffraction in structural biology, but only 2 of these chapters were about electrons. One of them, electron diffraction, Hua Chu wrote that, and this one on electron microscope imaging that Tim Baker and I did, because that's what we were focused on, trying to make it better at the time. And this just shows 4 examples of different types of specimen. On the left are images from Joachim Frank's work, where he was at Albany then, on E. coli 70S ribosomes. That was the best images they could get on film in year 2000. And the structure they got, which at the time was a landmark, 11 angstrom resolution, large subunit, small subunit, parts of long RNA and tRNA that they could identify by doing difference mapping. The second panel is hepatitis B virus cores that Bettina Botcher, who came as a postdoc from Germany and now is back in Freiburg, I think. And she was the first one to get a single particle structure by cryo-EM beyond 10 angstroms, so sub-nanometer resolution. And that structure they got as icosahedral symmetry, but T equals 4, so 240 subunits. And the protrusions there is a little 4-helix alpha helical bundle. And you can see the ridges for the alpha helix. And they were able, even though it was only 7 angstrom resolution, they were able to build a model that later on turned out to be more or less correct from where the polypeptide is arranged. And then the third panel shows that you can get other symmetries, including helical symmetry. This is Ron Milligan's work at 30 angstrom resolution by difference mapping, showing actin in blue, tropomyosin in green, and then different domains of myosin in the pink, orange, and yellow. And that told you a lot about how muscle contracts, how the myosin attaches to the actin and pulls the 2 thick and thin filaments along to give you muscle contraction. And then the last panel was one of the 2D crystals. In the end, there were only about a dozen electron crystallography atomic models made using electron cryo-microscopy. That was the second one. That was Werner Kuhlbrandt's work when he was a group leader at the MBL on the light harvesting complex from green plants. You don't see much contrast because the contrast is low, below the noise level, but you can find in the computed transform of the images, you can find the spots and you can find the amplitudes and phases. And his structure showed 3 transmembrane helices and about 13 chlorophyll A's and chlorophyll B. So they got an atomic model also in 1994. And so that was where things were round about the year 2000. And what was needed were hardware improvements in the microscope, which came from manufacturers. That's field emission guns, vacuums, stable stages, and so on. Alongside that, Jacques Dubochet, who had started out as a PhD student in Switzerland and was recruited by John Kendrew when he was director at the MBL to develop methodology in cryo-electron microscopy and specimen preparation. And they built a liquid helium microscope, spent a lot of money on it actually. And we actually used it at one time and got some good images of Bacteriodopsin. The impact of his work, the main impact has been this development of the plunge freeze method. And here you can see that it's a pair of forceps with a little 3mm grid on the end, and you blot your sample, plunge it into liquid ethane at the temperature of liquid nitrogen. And that was done by Alistair McDowell and Mark Adrian with a piece of filter paper and liquid ethane. Very simple. You just drop it down under gravity, or they had this spring elastic band powered. And of course, now you buy for 50,000 or 100,000 euros, you can buy a computer controlled version. in 1984 or 85, when this was starting, we didn't have any plunge freezing equipment at all in the lab. Soon had. And now we have about 7 or 8 commercial ones and probably 10 or 12 homemade ones. So maybe we have 20 of these apparatuses at LMB actually, which different groups use for different purposes. That was the Dubourchet, Adrian McDowell plunge freeze method, which is still used, although with many modifications. People try to improve the problem of the air interaction of the proteins with the air-water interface, which doesn't happen in crystallography because you have such big drops. One of the first structures, they published lots of beautiful pictures in the 80s, actually, of different samples, muscle, ribosome, viruses, and so on. But this one had a lot of impact. Published it on the front cover of Nature in 1984. It was Adenovirus, and you can see a few of them have been broken by the plunge freezing and the formation of the thin film, but many of them are intact. And there is a slightly interesting story about Jacques, because he was hired by Kendrew, and after 9 years, Kendrew had to depart, and Leonard Philipson from Sweden came in as the new director. And one of the first things that Leonard did was to get rid of quite a few group leaders, including Jacques, who was terminated. Then he produced this picture, and he was reinstated, because Philipson worked for his whole life on adenovirus. And then Jacques stayed there till about 1987 and then moved to Lausanne, where he's been until he's still there. And there's a new institute called Dubourchet Institute going to be opened in a few months' time, actually. But there weren't a lot of computer programs in those days, and it took them from 1984 to 1991. Phoebe Stewart was a postdoc with Steve Fuller, a different group. Jacques wasn't so good on the computing side, but between the two groups, they got this model 35 angstrom resolution, where you could see subunits, but not really any detail. And that was more or less state of the art in the year 1991. And then it took another, 20 years until Hong Cho at UCLA, who'd bought one of the new microscopes, but still recorded pictures on film. So he had 300 kV instead of 100 kV. He had much brighter sources, 1,000 times brighter field emission guns instead of tungsten, but still recording images of adenovirus on film. And you can see the detail in the new images is much higher. You can see a lot more internal detail in it. And when those images were processed by Hong Cho's group, you can see on the left, they get a beautiful structure at 3.6, 10 times the resolution, so 1,000 times as much data. And then if you look within the structure at the individual subunits, you can see you can resolve the beta sheets, 4.8 angstroms apart between the strands, and even some of the side chains, you can begin to see them also here. And then that was published in 2010. And that improvement from 1985, 1991 to that was entirely instrumentation and hardware of the microscopes. So field emission guns, stable stages, much higher vacuums, better data, but still using film. So then these new CMOS detectors came in. That was one of the biggest steps forward. We knew it was going to be better, but it turned out to have a lot of aspects that made it a lot better. So this is a plot of the detected quantum efficiency. 100% means you've recorded every electron that makes the image, and you've found out exactly where it is so that you can find its position both at low resolution and at high resolution. In film was about, you can see it's about 30% detected quantum efficiency. And these were the 3 new detectors 6 or 7 years ago: direct electron, Gatan, and FEI, now Thermo Fisher. And we were involved with the Falcon series, which is now Falcon 3, 4, and hopefully 5, and so on. And the detected quantum efficiency has now gone up to near 100% because you're counting the electrons now. So there's about a factor of 3 improvement simply due to the efficiency of the recording of the electrons. However, because these new detectors are fast detectors, they take pictures every 40 frames or 1,000 frames per second. If the image is blurred, and that was one of the problems early on, you put the beam on and the specimen moves either mechanically or due to charging. And so because it's a movie now with the new detectors, you could do motion correction. And it turns out, although you might be lucky, one image in 1,000 might not move the specimen, and you get a good image. With the movie motion correction, you can make every one almost perfect. And that gave about another factor of 3 or so from the motion correction. And then the final thing is that as you put the beam on, the initial first frame of the movie is perfect image of, an undamaged, non-radiation damaged protein. And as you irradiate it over 10, 20, 30, it degrades it, particularly at high resolution. And so you can do dose weighting. And those three things each gave a factor of 3 or 4. And so in the end, the overall factor is about 30 or 40 times better. This is an enormous improvement. So now where we are, there's still some improvements to come, but they're factors of 2 or 3. No more factors of 30 or 40 in any of the electron cryo-microscopy. So that made a big difference. Going back again. And I'm just going to show you now 2 or 3 of the structures that I felt give you the best impression of the power of all the new methodology, hardware, software, and computers that allow you to tackle structures that were impossible before this. And one of them here is work on the mitochondrial ribosome structure that Alexei Amunds did in Venki Ramakrishnan's group in Cambridge. And of course, Venki had come from Salt Lake City in the year 2000 and had been doing Xray crystallography on ribosomes and has a 2009 Nobel Prize work for that, shared with Tom Stites and Ada Yonath. But he'd always been interested in electron microscopy. And Georges Cherez, who also comes from Xray crystallography, came as a group leader in 2010 and spent about 2 years writing some new software called Rely On. And so by 2012 or 13, it was just about working. And so Alexei Amunds got together with Xiaochen Bai, who was a postdoc in Shor's group, and took images of this, like top left, which was, and Alexei had worked for a couple of years trying to purify mitochondrial ribosomes with the idea of making crystals. But after getting together between Shor's and Venki's groups and taking images like this and using Rely On, they were able to get, by a method of processing the images called 2D or 3D classification of the particles. And that was an intrinsic part of Rely On because Shor's had done this years earlier using the XMIP computer system from Spain. So he was a pioneering of this classification methodology. And what it allows you to do is that if your specimen, for example, isn't perfectly pure, and that was, Alexei could never get pure mitochondrial ribosomes, always mixed up with cytoplasmic ones, and some of them had fallen apart and so on. And this classification allows you to see that out of the 6 classes, they said, give me 6 classes, and the computer just looks and finds 6. One of them is cytoplasmic ribosomes. 4 of them are the mitochondrial ribosome. Some of them just a large subunit, but a couple of them fully intact with the large and the small subunit. And then one of the other features in Rely On and the other software systems, like, for example, System or CryoSpark, you can focus the refinement and the determination of the position and orientation of each of the particles in that image so that you can put them together into a 3D model. You can do focused orientational position determination and get then a sharp image, even if your structure is flexible, of the part you focused on. And so they did that for the large subunit and for the small subunit. And then they got really good maps by processing the same data in different ways of the whole ribosome. And a couple of regions of that map, just showing you how well the methodology works. This is a little bit of Watson-Crick RNA double helix, side view with 3 bases, and you can see the map resolution according to the mathematical criteria is supposed to be 3 angstroms, and the bases are 3.5 angstroms apart. You can see the density comes down and up really nice. So looking at the map makes you believe that the mathematical evaluation is correct. And then there's bottom left is a Watson-Crick base pair viewed along the axis of the RNA, and then 3 regions of protein, an alpha helix, and then 2 beta bends with top right what's believed to be a magnesium ion at this resolution, which was one of the few. Of course, there are hundreds of magnesium ions and water molecules now in the higher resolution maps. This is one example of sort of an impure, flexible structure that still cannot be done by any other method than the single particle cryo-EM coupled with the computer processing. A second structure came when Kelly Nguyen, who was a PhD student with Kiyoshi Nagai in Cambridge and had finished her PhD by crystallizing a complex consisting of 6 or 7 proteins with a part of the RNA, but it was a 5.5 angstrom. It was a low resolution structure. And Kelly was the one who took the initiative in Kiyoshi's group to move into cryo-EM. And of course, over the years, many people joined in that process, including Clements when he was there. But this was the first structure they published, which was the Trisnurp, just about a good enough resolution with lots of focused refinement that are able to identify all of these proteins and a large part of the RNA. And again, the spliceosome is much, much more flexible than the ribosomes. So people have been working on ribosomes since the 70s, actually, gradually improving the resolution. But the spliceosome looked like it was totally beyond. And the people who identified splicing as a biological function never thought they would see a picture of it, actually. So they were really thrilled by that. And then a little bit of that map from Kelly. That's on the left, PRP3, and that's an alpha helical stretch of one of the proteins. And then at the top right, a little bit of RNA double helix moving into RNA single helix at the bottom. And again, but the resolution is not quite good enough to resolve the basis, but it was good enough to build the model. So that's a second example. And now, of course, there's lots of the intermediates involved in splicing, not only from the group in Cambridge. Kiyoshi died from liver cancer about a couple of years ago now, quite sad. But Yi Gongxi and a group in Tsinghua, and obviously here now, Holger Stark and Luhrmann in Göttingen, who've been working on it for years, actually, which is one of the reasons Kiyoshi felt keeping Xray and cryo-M separately. But anyway, everyone's benefiting from that now. And then a third structure, which is the only one, just to show the examples of it. This is an image of mitochondrial inner membrane protein complex 1 from the group of Judy Hurst, who has been in Cambridge for about 20 years in another MRC unit, mitochondrial biology. She had been, again, purifying it, trying to make crystals. Another group in that same mitochondrial biology unit was Leo Sasanoff, who, again, he started with EM, found couldn't get crystals, it wasn't good enough, so he'd switched over to doing X-ray crystallography on thermophilic versions of complex 1. But Judy had been working on this for 10 years or so, and they'd got crystals, but they didn't diffract very well, and it wasn't the entire complex. So she brought it over around about early 2014, I think, or maybe 2013. And within 2 or 3 weeks, Vinod Kumar in our lab, Jaipan Ju in Judy's lab, had got this map at 5 angstrom resolution of the complex, which you can see is fully intact, different views in the image there. You can see at the top left part, contoured at a high level, there are these 8 iron sulfur complexes. And the way that complex 1 works in the inner mitochondrial membrane, energy transduction, you feed in energy at the top, NADH, reducing the topmost iron sulfur complex. The electron flows down into the membrane and reduces the quinone, and then there's a structural change that's still being characterized. And actually, Leo Sasanoff is the leader in this work. He's got the best theory at the moment about how 1 electron from NADH makes a structural change that pumps 4 protons across the membrane, charging the membrane up, and then used to make ATP with the ATP synthase. But in the membrane, there are 76 transmembrane proteins in this complex, and you can see them all, but at 5 angstrom resolution. At a lower contour level, you see the detergent with the lipid from where it's been extracted from the membrane. And with about another year's work, Vinod and Shaipen managed to get a higher resolution map at about 4 angstrom resolution, where they could identify from the amino acid sequence, comparing it to the density, all 45 of the different proteins. And of them, 16 core subunits, which are the blue ones in the middle, are very similar in structure to the earlier X-ray structure that Leo Sasanoff had determined in that same MRC unit by X-ray crystallography of a thermophilic species. And then the other 31 subunits, the red ones, many of them are smaller, and they're peripheral, and they help in some way. We don't know exactly, but anyway, they ended up with a model, and then Leo, that was bovine. Leo had sheep, and now there are lots of groups now in Tsinghua, Werner Kuhlbrandt in Frankfurt, and then Holger Stark and so on. So really the whole power of the methodology has been brought into the different groups who were interested in this. And again, without the cryo-M, I think it wouldn't work. And then I wanted to show one last one, which is not a new structure that can't be done by any other method. This is human hemoglobin, and we always thought wouldn't it be wonderful if we could show Max Perutz a structure of his hemoglobin that he worked hard for his whole life, done by cryo-M, but we didn't. Max died in 2002, so it was too early. But Miriam Koshai, top left here, was another PhD student at Martinsried with Radostin Danev, who was their supervisor, and Wolfgang Baumeister And she was the one who said, I think we should try hemoglobin. And we thought it was a bit too small, actually, because the smaller it is, the less the signal and the harder it is to find the orientation position. But they had this faceplate that Radostin Danev had helped to develop, so-called Volta faceplate. And these are faceplate enhanced cryo-EM images by defocused phase contrast with the 2D classes and the 3D model that they ended up with. And looking at the heme group, you can see the proximal and the distal histidines next to the heme group, 3.5 angstrom. Really, they ended up with a really lovely structure of it, and it's only 64 kilodaltons. So that's another example of the breadth of the methodology. So taking a step back now from these examples of structures that show you what the power of the method is, we can look at the overall impact of cryo-EM relative to all the other structural methodologies. And on the left, we have this table starting around 1998, where there were no atomic structures done by EM. There were people could build models, but there were no structures being deposited in the protein data bank. And then, there's one, this is worldwide, now it's grown, it's over 3,000 a year. And this year it'll be, 4,000 or so. NMR grew and grew and grew, and it was about a quarter of the structures early on, because there were things that couldn't be crystallized, but they tend to be fairly small. It peaked in 2007, and now NMR is still very valuable, particularly for low affinity ligand design, and the drug companies like it. But now the Xray has risen to, so now there's 10,000 structures a year, but it's more or less plateaued. That spike you see in 2020 comes from all the crystallographers who switched to working on coronavirus. You can see there's a deviation from this exponential growth, and that's from all the cryo-EM people who were also told they could only go into the lab if they worked on coronavirus. So then one of the things we like to compare is this ratio of the structures deposited in the PDB by X-ray crystallography compared to electron microscopy. And you can see, if you go back to 2000, there was 1,000 or 2,000 times as many every year deposited. And then when all the better hardware came in, it dropped to 100 or 200. And then when the new detectors came in, you see 2012, at that point, it starts to go down. And then this is on a log scale. So it's pretty much a straight line. And this number here, the first few months of 2022, there's 2.5 times as many still being done by X-ray crystallography and being deposited. Of course, many of them are, let's say, multiple versions of the same protein with different drugs bound. But nevertheless, it's 2.4. And we reckon if this carries on, we're quite interested, because this depends, it tells you how many microscopes you should buy, and it tells the companies how much they should be charging for it if they wanted to carry on. So one of the interesting things then is to compare this with the absolute numbers now on a log scale of the X-ray, and they all have this exponential growth phase, but usually only 2 orders of magnitude, so about a factor of 100, and then it plateaus because you've run out of things to make it faster. And this table top right tells you roughly what the doubling time was during the exponential growth phase. And in the cryo-EM, it's about a year and 8 months it takes for the doubling of that. And that's been the same since 2012. It's been doing that exponential growth over 10 years. So the question is, can that carry on in the same way that in transistor design, in integrated circuits and computers, they managed to carry on for 50 years, which is over, it's 10, 20 million-fold improvement in the number of transistors per square millimeter. And of course, DNA sequencing has gone faster than this in terms of the power. So the question is, can that happen in the cryo-EM? And then one comparison that we like to do is, because in LMB, in Cambridge, in UK, we did have the first Krios, and we had it 2013, and it was the only one in the UK. There were 1 or 2 others in Europe, not many. So we've looked at this same type of plot now, so that the PDB overall, that's the same plot I showed you before, where we're at, roughly speaking, 2.5 times as many X-ray structures as cryo-EM. But at the LMB, we still have a lot of people doing crystallography. They go to the synchrotron. We have 1 X-ray set still. But you can see we had a crossover in 2017. The lines are roughly parallel. So this number here now is about 3 or 4 times as many PDB depositions by cryo-EM at LMB than there is. And so we haven't plateaued yet. And we don't know how it'll continue. But we think it's safe to say that other people will probably encounter the same, let's say, value for money judgment. And so we think it's probably, the whole field probably okay to about 2028. Then we should worry about, faster detectors. Maybe you could do structures in a second rather than in an hour or something like that. So yeah, so all looks very promising. So I thought I'd show you a few more examples that reflect things that, we're particularly interested in, which does involve using the methodology for something other, for example, for drug design. So I thought I'll show you maybe 4 or 5 different structures for people who haven't done any cryo-EM before. This is one example which isn't particularly demanding or anything like that, but it was one of the 10 or 11 specimens that Vinod Kumar and I, probably 2015, 2016, we wrote a review, and many people had been sending us images saying, is this good enough for cryo-EM? And usually they weren't. They were usually really bad. But we wrote back and said, this is very promising. But here is another example of something similar in size and disposition. This is what you should be aiming for. So rather than doing that, we said, okay, well, let's take lots of pictures and put them into a review. And we did that, picking structures from about 4 or 5 megadaltons right down to 10 kilodaltons to show what the images should look like if you did it really well. And one of the specimens we picked was human erythrocyte catalase. But the funny thing that happened when we took images, they formed, instead of lots of pretty pictures, although we published one pretty picture, they formed these ribbons, top left. And that was what happened when you block the thin film of liquid with your 1 mg per mil solution of protein. You blot it to a thin film, and the thinner the better, you could get better quality images. But they form these ribbons because the catalase molecules interact with the air-water interface on both surfaces of the thin film. And we said, okay, well, what can we do to get rid of that? And obviously one of the things, you put a detergent monolayer on it, and we tried a dozen different detergents. One of the detergents gave images like this one on the top right, where the film becomes thinner in the middle. You see all the catalase molecules then, instead of being, as in the top left, they're all in the same orientation in the form of ribbons. They've now been flipped. The thin dimension of the molecule forms a little raft in the middle, and then round the outside are molecules with the same orientation as the ones without the detergent. So we said, okay, that was dodecyl maltoside. We tried lots of other detergents, but the one that we picked in the end, several of them worked, but Chapso was the one which changed A and B into C, and at 1 mg per mil, you don't see many molecules, but this is exactly the number you expect. 1 mg per mil is, say, 0.1%. You shouldn't see very many. So then you compensate by putting the concentration up. This is 20 or 30 mg per mil, so now you see lots of molecules, all different orientations, and images like this are exactly what you need. You don't need many of them to get the 3D structure. So when we processed images like that, but first I'll show you this one, 1 mg per mil, 10 mg per mil, 25 mg per mil, and you can see the concentration fits exactly what you predict from Avogadro's number and the concentration and thickness of the ice. And then the other thing you need is this plot of the angular distribution. This is a sort of a plot that Katerina Naidenova, who's a PhD student with Chris Russo, developed this way of plotting it. And the one at the top, which shows only one orientation, that comes from those initial ribbon type images. And then as we improve the lack of interaction with the air-water interface, we eventually get it until we have a really nice, fairly homogeneous distribution. And for this, you only need a few images to get hybrid. And then here is the structure, top left, 2 angstrom resolution. Here's the overall model, and here are 5 regions of the protein where we've only contoured the part that's been identified. So that's the heme group in the middle of the human erythrocyte catalase. That's the NADPH with the 4 phosphates all nicely resolved. And then we have a helix and then 2 regions of beta structure viewed in the 2 different directions. And in the model, if you look at the graph plot, the resolution estimated from the 2 halves of the structure without reference to a model is 2.2 angstroms. From a model comparison to the density map, it's also 2.24 or something like that. So it's a really nice structure, and it's supposed to come out very soon in a journal that Fujiyoshi runs, now called Microscopy, Oxford University Press. Okay, so a second structure that shows some of what people have been doing with it comes from the work Vinod Kumar initiated while he was in Cambridge before he went back to India to help them develop cryo-EM in Bangalore as the one lab that was into cryo-EM at that point. So it's collaboration with a group in India that make this enzyme. PAZ is phenylacetic acid degrading enzyme. And what they've done is collected 4 data sets with and without 2 different ligands. So NADPH again, or octanyl-CoA, and then the maps at the top, the gray is the density and the red is the difference density between the 2 maps. So just like in X-ray crystallography, you, with and without the substrate, you do the difference density, you find the ligand, and they can build a really nice model of it. And that was published a year or so ago. So that then led the way for drug companies, and a lot of them from 1990 onwards, maybe 30 drug companies, had been using X-ray crystallography to analyze their drug target site for inhibiting or activating a particular enzyme activity. And this came from Haran Jyoti and Pamela Williams at Astex in the Science Park in Cambridge. And they were the leaders in the Cambridge community to persuade 5 companies to buy into the Krios system. And in 2015, they persuaded Thermo Fisher to run a little facility. And they now have 2 Krioses. 5 companies pay for it. They get 1 day a week on each microscope. And then we help them a bit. So we get Saturday on one of the microscopes. The university gets Sunday. And then the second microscopes in materials science, they get the weekend that's there. So that works. And then the companies learn by talking to the people in the academic groups. And this was a project, this was the first one that they published in Drug Discovery Today about 2 years ago now. And they picked beta-galactosidase, which was a sort of model enzyme that we'd worked on, LAC-Z, the original operon. And there you see 3 maps with 3 different inhibitors of the beta-galactosidase that they were using. And this paper, they collected 60 data sets, so 1 data set per day. And that was 60 of the days that they bought into at, say, £5,000 a day, I think is what worked out roughly. And then at the bottom was a genuine one they were targeting as an anti-cancer drug, pyruvate kinase with 2 inhibitors found. And now these companies who were dipping their toe in the water 5 years ago, they've all bought their own microscope now. And Astex has a Krios and 2 Glacioses, and it's just a little company on the Cambridge Science Park. And then AstraZeneca, GSK, UCB Pharma, and then Hactares are the other ones. And that's still going well. And I think there are other collaborative initiatives. There's an industrial Krios also as part of EBIC on the Diamond campus. A couple of other ones. Chris Tate is a group leader in Cambridge who has worked on membrane proteins for his whole life, but started to work on G protein coupled receptors about 2005 with a targeted mutagenesis method to make the protein stable enough to make 3D crystals for X-ray crystallography. But of course, with that background, it was quite natural that he was ready, primed to get involved with cryo-EM. But it was actually Javier Garcia Nafria, who was a postdoc with Chris and is now back in Spain. He was the one who took the lead in making complexes of different G protein coupled receptors with the trimeric G protein complex and using the same faceplate that Miriam Kochai did. And this is an example of an image of this adenosine A2A G protein coupled receptor with NECA, which is a N-ethyl carboxyamide version of adenosine that's a really good agonist bound to make it in the agonist conformation with binding of the G proteins. And that's the image, the 2D classes, the 3D map, and the 3D model, and then a little bit blow up where you've got the difference density from the NECA. And then a second one from Chris's group also is one of the serotonin receptors with donatryptan bound to it. And again, that's a cryo image, the 2D classes, the resolution plot, and then various bits of the map showing examples of the resolution. It's something like 3.6 angstrom resolution. And donatryptan itself is not a drug that was approved for human use, but one of the, in the same family, sumatriptan, is one of the anti-migraine drugs that you can either by mouth or by injection. If you suffer from migraine, that's one of the treatments that you can get now. So these are genuine examples where cryo-EM has become useful, but I don't yet know of any actual real drug that's in the real world, in real clinics yet, because it takes such a long time to get it approved. Okay, let's finish by showing you a little bit from the coronavirus, because this has had a a big impact on humanity. Hundreds of billions of money have been used in economic depression, stimulating the economy. And we even received, some of us who have an American, we received a check from the president to help us. Anyway, this is the SARS-CoV-2 virus that you'll all be very familiar with. But on the left are two drawings from Wikipedia, which are really distilled from all of the different illustrations people have made, showing the different proteins, about 20 proteins. But in the membrane, you've got this spike protein, that's the red one that sticks out. But on the right is an example of a group that has done some electron cryo-tomography. And that was one of the things I omitted in that early examples of what cryo was good for. But you can also use exactly the same equipment and then do, without doing any averaging, you just do a tilt series, say -60 to +60, and get a reasonably good map of individual particles without any averaging. And so in that top panel are cross-sections through the middle of, obviously, the formaldehyde inactivated SARS-CoV-2 viruses from John Briggs's group when he was in Cambridge. Now he's in Martinsried. And then you can see the spike protein sticking out, and it has various conformations: closed, open, 2 of the 3 domains in the trimer open, and then a fusion domain as well. So the cryo-EM has been used to characterize and give you real-life examples that allow you to correlate it with the model. And actually, one of the things I found out recently was that in the early years of coronavirus naming, there was a young woman from Scotland called June Almeida who was working in London, and she named these coronaviruses when she was an EM technician looking, and found these ones with these spikes sticking out. Obviously, it wasn't a pathogenic one, it was just a common cold one at that stage. And then a second slide about SARS-CoV-2. And of course, there are thousands of structures deposited now from different groups. But this particular one comes from Yao Kong's group at Tsinghua, and they studied by single particle cryo-EM the spike trimer alone, and then the spike trimer in complex with the receptor binding domain of the angiotensin converting enzyme on the surface of the cell that's being infected. And then there are lots of others with and without different antibodies and so on. And that, in principle, should help you in figuring out what are the best parts of the structure to use for making antibodies, for making vaccines and so on. Or if you wanted to make an antiviral inhibitor for that interaction. And then the 3 drug targets in the coronavirus are the RNA-dependent RNA polymerase, which structures, lots of them have been done by Xray crystallography and by cryo-EM, with and without the different drugs, including one from a group in Cambridge, Chris Russo, Katerina Kaidanova again. And then there's the main protease of the virus that cleaves up the polyprotein which the virus needs to live. And that is a little bit too small. It's only been done by Xray crystallography, but one of the drugs, Paxlovid, again, is done by structural biology by Pfizer, who had earlier drug targets on related proteins from similar viruses. So that gave them a head start. But lots of companies have been targeting this because of its importance. I think I've given you then a little bit of what I thought were the main factors that led to the cryo revolution, how well it's going on, and how we expect it to carry on for at least 10 years. And then we should worry, after all, how do you make it faster and better and more accessible to people from different backgrounds who are not necessarily experts in it. So I just want to finish by looking at what do we expect from electron microscopy. So one of the things we're focused on in Cambridge at the moment is how to make it cheaper so that you don't have to pay the 5 or 6 million for the Krios. And we thought 100 kV with a field emission gun and detectors. And, understandably, the companies JL and Thermo Fisher that sell the microscopes, when you say we've got this great new idea to make it 10 times cheaper, they're not full of enthusiasm for this idea. They say, why do you just keep buying more of the expensive? Anyway, we're trying to do that, and we found a little company in York in the UK which has been making field emission guns for scanning EM, so low voltage, 20, 30 kV, but they've now made one that's suitable to go up to 100 kV, but not 300, but we think there's other advantages in 100 kV. Radiation damage is a bit less. And then the detectors are all designed for 300 kV for high voltage, and they work better, 300, 400, 500. But as you come down, the electrons, they scatter around and reduces the efficiency. So we need new detectors. And there are a number of projects now to develop detectors that are focused and most useful for 100 kV. So once the cost comes down, maybe every group can have their own, and then they don't need to worry about booking ahead and waiting for 2 months, which is what we have to do at the moment at Cambridge. Another direction is that now that we know how to identify the position and orientation of molecules from a purified preparation, maybe you don't need to purify it. Maybe you can just put membranes in that haven't been solubilized, haven't been purified, and identify by this, let's say, called motif searching, which Nico Grigoriev now at UMass has been developing, but with tomography in mind. But it doesn't need to be tomography. It could be thin films on carbon films and surfaces and so on. So it could well be you could get high resolution structures directly using unpurified or partially purified, because now that we know what we're doing, you don't necessarily need to look and see and know that everything that you see is the same thing. It could be a mixture of states. So we think that both in terms of affordability, accessibility, and the range of specimens that you could use it for, there's a lot of unfulfilled growth coming up. So we don't see any plateauing of the value of it in the future. And then meanwhile, all the synchrotrons are being upgraded. And the only one so far that I know is ESRF has upgraded to finer beams, brighter beams. And that's made X-ray tomography much more able to be done in a reasonably short period. And there's one group in London, Peter Lee, who's been going to Grenoble, and they've been able to get, because X-rays penetrate much deeper than, for electron microscopy, you need a specimen maximum a few thousand angstroms. But for X-ray, you can go up to much thicker specimens depending on the energy. And they've got a way of doing a tomogram of the whole tissue, say a whole lung, and then they can focus the beam in just on, let's say, a centimeter or a millimeter square and get a higher resolution picture of a little part of the structure in a reasonable time. And so that, together with upgrades of the other synchrotrons, and Diamond, for example, are studying what to do, but it's not going to be online for about 8 or 10 years, actually, so 2028, 2030. So there's scope there. And of course, obviously, the crystallography beamlines, they can keep on getting faster and so on. And then there's a lot of interest also in both protein folding and drug interactions, where you evaluate the energetics of it in the computer. And people have always been enthusiastic. 1985, I remember the computational people said, oh, we don't need structure anymore. But soon that disappeared. But it is better now. So if you do drug design and bring into it computational calculations, instead of having to test 3 million compounds from your library, you can manage with 1,000 or so, and the hit rate is much higher. But if you get the hit rate to be 100%, then you would have said, okay, we've learned as much as we need to about the energetics. But at the moment, the energy functions are not quite accurate enough. So I think probably we'll need lots more experimental structures. But eventually, you might well find that there's a parallel between. And this is not the pattern recognition from AlphaFold and RosettaFold. This is genuine using the energetics to actually identify where are the binding sites for drugs and so on. Okay, so that's a little bit about the future. So let me thank everybody. So I mentioned at the beginning, I'd still be doing X-ray crystallography, trying hard to make crystals and so on, and succeeding some of the time if I hadn't met Nigel. And we had that early work at room temperature electron microscopy, not cryo, and then spent a long time with collaborations with a number of groups. So Ken Downing and Bob Glaser at Berkeley, Fritz Zemlin and Eric Beckman in Berlin, and Jean Le Paul, who was in Jacques Dubochet's group. They had a liquid helium microscope that they developed by themselves in collaboration with Siemens at the MBL. And data from all three of those microscopes helped us to go from 7 to 3.5 angstroms over a long period. And Tom Seska and David Agard and Joyce Baldwin were doing that work, going from low resolution to high resolution with bacteriodopsin. And then we switched to single particle EM and trying to develop better detectors. And Renato and Nicola are two Italian CMOS chip designers. And Nicola is still at Rutherford Appleton, right next to Diamond, designing next generations of both 300 kV and 100 kV detectors. Renato has his own company now in Barcelona, But again, we're still collaborating with them with another project for 100 kV together with Thermo Fisher. And then Greg McMullen and Wazi Faruqi are in our MRC lab helping to develop the detectors. And then Shausha has been involved with a lot of these. She was the only cryo-EM system manager when she was recruited in 2003 from Birkbeck and Imperial College. But now she has 4 other people. So we have 5 people running all the microscopes, because lots of people who have no background want to use it, and then they need help. Whereas before it was people who knew what they were doing and so on. And then the people involved in our switch from electron crystallography to electron microscopy, Sriram and Jacqueline Mill came on a 3-year sabbatical and then went back to NIH and now are in Vancouver. Nico's at UMass. He was there, wrote Freeline and now System. John Rubinstein was a PhD student with John Walker and did one of the first ATPase structures at 30 angstrom resolution, 2003. Now they have lots of them at high resolution with multiple states and so on, and lots of other groups as well. Peter Rosenthal did a lot of the work analyzing what the problems were and how to get, and he's now leading the cryo-EM in the Crick Institute, and Vinod Kumar in Bangalore in India, leading the EM, cryo-EM development in India. And then most recently, Chris Russo came initially as a postdoc with Laurie Passmore, and now as a group leader, trying to focus on problems like air-water interface, how to stop beam-induced motion and so on, and to make the method much more powerful. And then the last one is to just say, I've of course shown some of the work we've been involved with, but Adenovirus from Liu Boche and Hong Cho and Phoebe Stewart, a mitochondrial ribosome, the Trisnurp, mitochondrial complex 1, hemoglobin, catalase, P. So these are the structures I've used to kind of illustrate a little bit the development of the method. 2 GPCRs, which Chris Tate's, that's now entirely Chris Tate's group, and then work from the rest of the world, a little bit from LMB, in terms of SARS-CoV-2, showing you that it's not just has long-term impact, but it's also topical impact. So anyway, hopefully that's given you a bit of a flavor. Thank you very much.